DTNBP1. DTNBP1 stands for dysbindin. Alternative names for dysbindin are biogenesis of lysosome-related organelles complex 1, subunit 8, BLOC or block 1, subunit 8, dysbindin 1, dystrobrevin binding protein 1, Hermansky-Pudlak syndrome 7 protein, and HES7 protein. Dysbindin is a component of the block 1 complex, a complex that is required for normal biogenesis of lysosome-related organelles, or LRO, such as platelet-dense granules and melanosomes. It plays a role in synaptic vesicle trafficking and neurotransmitter release. DTNBP1 also regulates cell surface exposure of DRD2. It may play a role in actin cytoskeleton reorganization and neurite outgrowth, and may modulate the MAPK8 phosphorylation. This gene promotes neuronal transmission and viability through regulating the expression of SNAP25 and SYN1, modulating the PI3 kinase AKT signaling and influencing glutamatergic release. DTNPB1 regulates the expression of SYN1 through binding to its promoter. It modulates prefrontal cortical activity via the dopamine/D2 pathway. The biological processes are actin cytoskeleton reorganization, enterograde synaptic vesicle transport, melanosome organization, neuron projection development, platelet-dense granule organization, positive regulation of neurotransmitter secretion, regulation of dopamine receptor signaling pathway, enterograde axon cargo transport, blood coagulation, membrane organization, neuron projection morphogenesis, positive regulation of gene expression, post-Golgi vesicle-mediated transport, and regulation of dopamine secretion. Hermansky-Pudlak syndrome, also known as HPS, is a disorder characterized by oculocutaneous albinism, bleeding due to platelet storage pool deficiency, and lysosomal storage defects. Steroid storage in the lungs is associated with pulmonary fibrosis and is common in HPS. Defects in DTNBP1 are associated with susceptibility to schizophrenia. Genetic mutations lead to alterations in the glutamatergic transmission in the brain and modified AKT signaling. Protein levels and expressions are reduced in nerve terminals of the hippocampus, and there is an increased release of glutamate in schizophrenic patients. DTNBP1 also plays a role in synaptic vesicle trafficking and neurotransmitter release. It also plays a role in the regulation of cell surface exposure to DRD2. If you have any concerns about dysbindin, please contact a practitioner who understands epigenetics and nutrigenomics.